So we are going to start the second session for today, and this will be the last session for this Gyan course. So this is on the case study 2. So for the case study 2, it was about developing a microbial protein, a protein that can be produced by the microbe, or which is actually produced by the microbe and is utilized in the industry on a wider scale. So the one which I gave you as an example, or not as an example, I wanted you to work on would be alpha amylase. So alpha amylase is produced by the microbes. The idea was you're going to use the techniques that we had discussed as part of the synthetic biology to impart properties into alpha amylase that will make it much more viable. Alpha amylase is not a new enzyme. It is widely used in the chemical industry. It is widely used in the pharmaceutical industry. It's widely used in the food industry. So it has been in the market for a very, very long time, but there are issues which are still existing with respect to its production, with respect to its quality to a certain extent. I will not say to production. Production has been always been optimized for majority of these products, but quality does matter to a certain extent. And our intent was to come up with a newer solution using precision fermentation. Now, the definition of precision fermentation is where you're using something which requires recombinant DNA technologies. You're producing it in a microbe on a larger scale, making it much more viable. Correct? So I'll start the same process the way we started having the conversation in the first session. So we'll start with the group or the person who submitted the case study first. Mr. Rajesh?
Yeah, I will start.
So what we have for the case study 2 for Mr. Rajesh is again the same company, Swiss Shift. You're not changing the company name? Okay. So it's a second product from your own company. Cool. I like it. So you have diversified, which is a very, very good thing. You know, like the idea of alpha amylase, like many people have obtained alpha amylase from soil. It's like it's an enzyme which is produced by the microbes when they're in the worst condition possible. Like if they need to just survive somehow, like they're clinging to death, hanging from the cliff, like that type of situation exists for them. That's when they start producing this enzyme, and it's actually very beneficial for them, for their survival. And that's the only reason why we are looking at it, because it's a prominent enzyme, it's widely used. There has to be something new that we can add to our conversation or through our conversation. Do you have any questions with respect to the case study 2, like how you would have approached, or no, not case study, case study 1? Till the time they fix the technical issue, we can go there. If you don't have any question, Mr. Rajesh, if you can explain what exactly you did and how you approached it.
Good afternoon, one and all. So I am with my second case study, that is developing a microbial protein, alpha amylase. Okay, so where we are coming with the same team, I'm in Sweet Shift. So here I went on coming to the procedure regarding the conversion, making this. So then starch is the one of the best feedstock for this one. So the country like India, we are having huge amount of starch as per the reports or something. So 17% of world's agro residue is having there in India. So I read with the different papers, they are using every residue. So like potato or like wheat, barley, corn. So and whatever the residue is there, they made different. I mean, they used all these agricultural residue feedstock, and they tried by using these bacteria one I mentioned, bacillus, this one. So and they changed the gene modifications with respect to that one and made. Recently, I read one paper in 2024. I think that one I read about that one. So where I selected uh, these here bacteria which can, because which can give the rapid production or for the enzyme production it will be helpful, like that. That's why I selected. And the gene modification and where the gene cloning, where these the parameters are there that I am presenting. So giving the carbon nitrogen sources, and here the temperature we are
So this is
restricting
for your
for
the
this for
for the
this
fermenter.
ferment.
Okay.
Yeah, for the fermenter, the temperature source 37 degrees, and the speed also mentioned. So some mentioned 250, 260 like that. So as in medium like that, I went with 180 to 220 RPM.
So this is something that you decided upon.
Yeah, it is
Okay.
said. So they went for 250 to 150 to 250 like that. So I opted it like this is, and the pH value 6.5 to 7.5, and the dissolved oxygen 30 to 50 percentage, and the carbon sources will be the glucose, and the nitrogen will be the ammonium salt. Where here for this, if you went with the exterior tank, so this will the best one. So then I selected for the batch fed batch reactor. Even people did with this reactor. So and where the main reason is we can add the nutrients as in this one in the substrate. And here the purification, I mean the extraction process includes the intercellular setup, where the cell lysis, protein extraction, purification, and the concentration, this can be there, through which the alpha amylase can be obtained. And coming to the techno-economic analysis, where so the same I mean previously we discussed about in the plant protein production. So there the same methodology I followed. The raw materials, and the where the cost bacteria and the cost of starch and the enzyme modification. Here the enzyme modification cost reduced because people did on this when compared with that one. So that's why I reduced some cost in this, and the energy cost for the aeration and the stirring and heating are there, and the purification where the filtering and centrifuge, and the capital cost which includes the reactor and accessories and space infrastructure, and the labor cost adds um, the same like skilled and semi-skilled. Uh, so here the thing is total cost budget that I proposed. Oh, sorry, the it's a blunder. Okay,
I didn't mean to disturb you. So
yeah,
continue.
sorry. And the total cost where which is actual, if you write a proposal with respect to small one batch like proposal, it can cost of 10 lakh. Where here for India, the country like India, these as I said, we have huge amount of the agricultural based residues which can be available, through which we can use this generate this alpha amylase protein. So and the potential applications are also much more when compared with like these food beverages and pharmaceutical and the bio type detergents, and these animal feed some mentioned, but like that. So here so that's why so when compared with the others, so alpha amylase production is done huge in India, so it has a fruitful one. So anyone can do the change in the gene and they can run it.
Okay.
Yeah, and sorry for the mistake, that is
That's okay. That's okay.
yeah,
Even that was there in the
yeah.
previous one.
And
So.
this was preliminary survey that we did with respect to some papers.
No, I'm glad that at least you had an opportunity to go do the survey,
Yeah, yeah,
right? So at least you got to know that there are certain things like monolin and alpha amylase that you
yeah,
can look
obviously.
at.
Yeah,
As an entrepreneur yourself, you should have broaden your
the
scope.
mind.
Cool. Uh, One thing that I would like to bring to your notice, like alpha amylase, as I said, is produced by the microorganism in a worst case scenarios, like in the worst case situations of it. So the question to you would be, so when we are looking at the feedstock, we had conversation a couple of days back about the paralis and all the burning of the paralis in Haryana and Punjab. Do you think setting up a small scale industry where you can actually collect all of these feedstocks, break it down, because you have to do some sort of pre-treatment to the feedstock. You cannot use the feedstock as is, right? I cannot use the stalks of the plant
Directly.
into the fermentation system. So what about the pre-treatment? Because I haven't heard about pre-treatment from any of the groups till now.
Yeah. So basically, from 2019, I am working on the agro-residue management.
Okay.
So through which I started my startup also. So even so the thing is, so collecting the residue in a clear way when I am going to it, collecting the residue from the farmers is a big challenge, because if you want to collect, you have to start this one in before their before their crop growing time. So before in the months of June, July itself, we have to talk with them such that you keep your residue aside and like that. So then what we will do means, and we have to monitor their production. And before they are setting their harvesting time, we have to collect the residue and we have to keep them in a host zone like that. I mean, we'll call it as an buffer zone or like that, which is of 50
Wait. So you are
kilometers.
saying you're going to collect the residue before the harvest?
After
After
the
the
harvest,
harvest, okay.
after the harvest, before itself, we have to go and talk with them that don't throw
Or
it
you're
out.
making the arrangements with the
Arrangements.
farmers. Okay.
Yeah, arrangement for the farmers, and we have to collect them and have to keep them in then stock or go down, which is in like a mandal, right? Like mandal type, that is 50 kilometers max, 25 to 50 kilometers of that one. So there we can set up a small go down. Okay, so that is the stock point. And with respect to the stock, we have to make one arrangement. So to such that the stocks are within a range like that. So such that if you keep with respect to a mandal, so usually the mandal will be of this much size only, between 50 kilometers. We can get that. You can keep a stock point. So then after this, then the pre-treatment process, there you can establish your warehouse through which you can run it. So the same model we proposed in our district, and even I am following the same model for my startup.
Can I add to that?
Yeah, you can.
So Food Corporation of India has godowns, has the space, has the facility. Why not utilize what you already have?
So
Why do you
actually,
have to build a different warehouse?
so actually food processing, this one is there,
Food
godowns.
Corporation of
Food
India.
Corporation of India godowns. So there will be not, if you see for a district, they won't keep one or two for a district. As for my district, I am from my district,
Okay.
district. My district from Andhra Pradesh. So my district has three itself. Okay, so which are in that in this range only, 50 like that, and they will think and they will charge actually for this.
No, what if the government is also interested in that? I don't think government will like to bill itself for doing something beneficial for the farmers. I'm just proposing. I'm just giving you an
Yeah,
idea. You can
maybe idea, good
initiate a
idea.
conversation with the MLA and the MP of that area and have a conversation about FCI. But coming
That's-
back to your
thing.
thing, any questions for him? Okay, if they don't have any questions,
Thank you.
I have only one single question. You're going with the same process of cell lysis, protein extraction,
Yeah.
purification. My question to you is, why do you want to purify alpha amylase to a pharmaceutical grade level? Why can't we just use it or keep it up to the food grade level? Because food grade level is not that high. You don't need that much of purification, and you might be able to make it cheap as compared to the pharmaceutical grade.
Based on the, I don't know that answer, but I am saying as
No, I'm just suggesting. So, if that is a possibility, let's say, will you be interested in doing that? Like reaching only up to the food grade level quality?
obviously food grade will give the more because
Will it give you a bigger market as compared to the pharmaceutical grade?
currently pharmaceutical will get more margin. Suppose in small medicine also they will take more.
You're talking about money. Nice.
Yeah.
You're all thinking about the dollar. Sorry, not the dollar side, the rupee side.
Indian rupee.
Yes, good. I appreciate that.
Thank you.
Thank you. I like that entrepreneurship in you. But if you're really interested in these type of businesses, you seriously need to learn a little bit more biotechnology. So gene cloning and
Knowledge
how you're
things.
doing the recombinant DNA technology, or at least hire somebody from the biotechnology group
Yes, sure,
for your
sir.
company.
Actually, I missed the same for this one. Even I applied for the BRAC, okay, in 2020 itself. So there I got to know that I missed in 0.14. So I never heard about that much after
Okay,
0.14
good,
values.
good, good.
So
I will.
0.14 I missed the project. So hope
That's
next
fine.
future
That's
I can
fine.
join. Bilal.
No, but see, there are the reasoning is that when you're looking at feedstocks just like that. For example, in the previous session when we were having the conversation, we talked about the monoline protein if it is added in the food product itself, and then we can definitely do pasteurizations and things like that. I think it was the third group, Bluff, right? That you suggested that. But when you are doing like because for the pasteurization, you're going to use heat exchangers. If you have protein in it, it might change its structure, and that might lead to flocculation, and your efficiency of your heat exchanger will reduce. So what will you do? It can even denature your protein, but let's say if you are able to keep it in the starch, maintain its structure and everything, but there might be a situation where it will interact with other components of the food, because you don't know like how, the protein and starch relationship, right? So when you heat them at a very high temperature, what sort of reaction happens? Maillard reaction, right? I'm sorry? It's more than 100 degrees Celsius? Yes. Okay. Okay. Good. Any questions for Rajesh? There is a question at the back, please.
Uh, uh, so basically, what are the gene modifications that have been considered for this particular experiment? You know, like you have thought some like there is some kind of idea, right?
At least whatever you have studied in this particular course, you could tell that. If you want to, like, you want time to go back to your notes. See, I think, see, for alpha mileage, you would be looking at the same. You take the microbe that actually produces it. You don't have to, sometimes you don't even have to do the gene modification for it. You take the microbe that produces it, but you might take the genetic DNA and insert mutations so that you might get the alpha mileage with the specific properties of interest. That is a possibility. That would be the approach that I would like to take, because that would be cheaper for me rather than taking out the gene of alpha mileage and then growing into something else.
Yes, sir.
But I will choose the organism that actually produces it. Just requires the enough carbon source, but I will definitely like to initiate some sort of mutation in it to get the enzyme of my interest. Heat stability is a very important factor with respect to alpha mileages. The alpha mileage which is widely available in the market, they are not very heat stable. So that sort of mutation is something that I'm expecting.
But sir, there are like vectors now available, like you can say like PUB 110,
Yes.
which provide like which also contains like initiation signal, like ME signal that can promote more production of the required protein of interest, which
Oh,
is
yes.
alpha amylase.
See,
So,
production itself is not an issue. Like, okay, if you want to produce a lot, it's fine.
yes, sir.
That's if that's the only requirement. But if I want you to produce it, but I want you to have a specific property attached to it, if it doesn't have that property, then it's not valuable to me. Right? Enzymes, because by virtue of their functionality, right? By virtue of their functionality, are important. If they are not functional, they are useless. They have to have a specific function to it, right? A mileage has to cut. But if it is not heat stable, then it might not be a viable option for me. If my application requires it to be heat stable, I'm just putting out an answer for him, This is not my answer alone. I'm just putting up an idea there. Any other question? No? Then in that case, we will go to the team procatalysts. Should I change the slide?
So good afternoon, one and all present here. The case study 2 is developing a microbial protein alpha amylase. So alpha amylase is a starch hydrolysing enzyme which breaks down alpha-1,4 glycosidic bonds. Alpha amylase is used in various industries like food industries to convert starch into sugars. They are also used in textile industries, detergent industries. So it concludes that alpha amylase is a very important industrial enzyme. And for producing the alpha amylase, we have selected a bacteria, namely Bacillus alkalophilus. And the reason for choosing this bacteria, basically, it produces a stable amylase protein which stands up to 55 degrees Celsius, and it is an alkaline alpha amylase. And the vector the host we are choosing is again the Bacillus subtilis, WB600 strain. And a reason for choosing this strain is basically in this strain there is a knockout of 6 genes, and those genes are basically responsible for producing the protease enzyme. And the protease enzyme basically degrade the recombinant protein. And in this WB600 strain, those genes were knocked out. So we can prevent our protein from degradation, actually. So the process goes like that. And first we have identified the gene. The gene sequence was given there. It was not the full sequence. I have only taken a small part just to prevent the space. And after that, vector and host selection was done. Vector was taken as the PMA5, which is an shuttle vector. It can replicate itself both in E. coli as well as in subtilis. And the host chosen is Bacillus subtilis WB600 strain. After that, the primer was synthesized for the specific gene sequence. And in that primer, there are restriction enzyme coding sequences also, because we want our gene to be specifically ligate to our vector. So we have incorporated 2 restriction enzymes, the BAM1 and SAL1, BAMH1 and SAL1 gene into the sequence. And then it was amplified using the PCR through at standard procedures. And after that, both the gene and vector was digested with the restriction enzymes, the same restriction enzymes. As it contained the sequence, so it will ligate to it only. Then ligation reaction happens, and then selection was done to differentiate between transformants versus non-transformants. It was done using 100 microgram kanamycin per ml and 1% starch. So 1% starch is used because if our transformed cell has amylase gene into it, it will basically hydrolyze it. And we can confirm that these colonies are having the amylase gene and these colonies are not. So by picking up the transformed colonies, we basically cultured and gone for the pilot scale fermentation process. Change the slide, please. So this is the vector design. As you can see, our alkaline alpha amylase is between the 2 restriction enzyme sites. We have incorporated the sticky ends and it was ligated into the vector. And the right hand side figure shows it is basically a SDS phase diagram in which जो arrow है वो basically 56 kilodalton protein in which confirms that our crude has the alpha amylase into it. So it covers the upstream process, and the downstream processing will be explained by my partner Vishal. Thank you.
So after incorporation into Bacillus subtilis, so the mode of fermentation that we have used here is solid state fermentation instead of submerged fermentation. The reason being it will have low moisture content, better product recovery, low water output, and lack of foam built-up. The carbon source we have used is the substrate that we have used is wheat bran, which is an agricultural waste. And then the nitrogen source we have used ammonium sulfate, inorganic source. In the study, we have found that the pH, the optimized pH is 7.1 for the production of alpha amylase using the Bacillus subtilis, and temperature is around 37 to 40, but on a study we found that the highest amount of alpha amylase production is around 40 degrees Celsius. And the bioreactor that we have used here is tray bioreactor. Now, sir, can you change the slide? Now the downstream processes include extraction, filtration and centrifugation, precipitation, dialysis, purification, concentration, enzyme activity assay and storage. So for the extraction purpose, it is basically used to recover the enzyme from fermented substrate by using an appropriate extraction buffer, example phosphate buffer or tris-HCl to the fermented material. A solid to liquid ratio is maintained, that 1:5 to 1:10 to optimize enzyme recovery. Okay, so after that, after getting the alpha amylase by shaking the mixture to facilitate the release, we can use filtration and centrifugation. Obviously, filtration, we use coarse filtration to remove the large particles, then centrifuge it to clarify the supernatant. After getting the supernatant, for the precipitation and purification process, precipitation is done. We use the crude enzyme for a 0 to 30% cut. We will use the precipitation dialysis process, which include the like 0 to 30%. We will add 268 mg if into 10, that is 2.68 gram of ammonium sulfate. Then the stir the mixture for 30 minutes. The solution is centrifuged, pellet is taken out, supernatant is kept aside, and the pellet is again resuspended in the phosphate buffer. The supernatant that is kept is again used for 30 to 70% cut, and again ammonium sulfate is used, and again the dialysis is performed. After this, the purification, the data that we get, so can you go to the previous slide? The data that we get is protein concentration is 0.17 mg per ml. The extract activity was 2.243 micromole per ml per minute, and the specific activity was 13.19 micromole per minute per mg for 48 hours of extract. Now, for the purification, enzyme activity assay, we can use the DNS method for enzyme activity assay, and for storage or the SDS page also. For storage purposes, we can use stabilizers like glycerol or sorbitol. For short term use, we can store into 4 degrees Celsius, or for the long term, we can use lyophilization or like freezing.
So you are generating a powder.
Yes. And the cost economic analysis will be done by my friend.
Good afternoon. So the first step will be the clone of the amylase gene into the host organism Bacillus subtilis WB600 strain. So we found out the approximate the value of the for the gene editing was $5,000 to $10,000. So the second step will be the media formulation, where we are using the carbon source as the wheat bran and nitrogen source. So we found that for the 100 liter of the batch culture, we have to spend almost $12,000 to $200 for the 100 liter of batch. And the inoculum preparation for the seed fermenters was found to be $500 to $1,000 for the seed fermenter preparation. Since we are using the tray bioreactor, it was found that the tray bioreactor will range from the $10,000 to $30,000 as per our need. For the as we know that the cheap one is also has the manual control, as well as the expensive one have automated manual controls. So the fermentation process includes the temperature control, pH control, and agitation control by including all the expenses with the labor and electricity energy consumption. We have concluded the approximate value up to $2,000 to $5,000 for the batch for the fermentation. So the cell harvesting comes with to separate the biomass from the fermentation broth was found to be by the centrifugation. by using the agitation speed and energy consumption, we found to be $1,000 to $2,000 per batch. And the cell lysis, which was done by the high pressure homogenization, so we have to find out the most proficient one, which was found to be up to $1,500 for the instrumentation, which was the one-time cost. So the purification, as Vishal said, the purification includes the ammonium sulfate precipitation and dialysis, which is quite expensive, which concluded up to 5,000 to 8,000 approximately dollars for the per batch volume. For the quality assurance, we are using UV visible spectrophotometer, which was we have to spend one-time cost for that spectrophotometer, which was found to be around in between 750 to 1,500 for the quality assurance. And ultra filtration for the 4 high grade, we have to do the ultra filtration to remove the salts and concentrate the proteins. It was found to be $1,500 to $2,500 per batch. And drying and packaging, which was done by freeze drying and lyophilization, or the lyophilization in the powder form for the storage for the convenience of the transport and the storage condition, it was found to be $1,000 to $3,000 per batch. So we estimated up to our one-time capital with including the labor, energy cost and everything. We was found to be our yield was up to approximately 10 to 50 grams per liter. So we have to find out the exact value so we can sell out. It was found to be $1,000 to $5,000 $1,000 to $5,000 per kilogram of the purified alpha amylase for the food grade quality. Thank you.
You said food grade quality
Yes, sir.
after dialysis and all.
For we are doing the ultrafiltration for that.
Ultrafiltration and dialysis, you get food grade quality. You are getting the best grade quality actually.
Pharmaceutical, sorry. Amylase, sir.
Okay. Any question for the group? Don't be hesitant. I think you all have had like a meeting. You don't ask me, I don't ask you. Okay, let me ask them. Okay, why solid state fermentation?
It will lead to low cost because downstream processing is a little bit easier. Low moisture content we can maintain in the submerged will have high moisture
Okay.
content.
And the pH of 7.1. If I may just wait here for a second, you said pH 7.1, 6.5 to 7.5. So it's-
Actually, I referred to a paper where there was a study being done for beta subtilis, and it was measured for the pH optimization and temperature optimization, and they got the temperature 7.1 around 7 and temperature around 40 degrees Celsius.
Okay. Not a bad idea. This would be solid state fermentation is cheaper, it's easy to maintain, easy to clean. But the question would be, you said in your, I'll say, economic analysis, not the techno part, economic analysis, you're spending $10,000 to $30,000 for this large scale fermentation. That's a one-time capital expense. Is alpha amylase the only product that you're going to have out of that fermentation system? Year after year, day after day, month after month, sorry, day after day, hour after hour, day after day, or week after week? Or you're planning to produce something else also? Because $30,000, if I have to spend, I would like it to be versatile enough.
Sir, we're actually planning to use 100-liter batch. It
Okay.
maybe comes in a pilot scale. So here we can do multiple product facilities where we can produce multiple products. Like if you see the process, it is actually same for the previous case study also. It also same for this case study also. So this, and I have seen the many industries developing this scale for facility for testing the many products before going to production scale. So in small changes we can use it for a multiple product facility. Yes.
See, 100 liter is a pilot scale.
It's pilot scale, yes.
the reason I'm asking this question is because I personally didn't teach you the scale up of solid state fermenters. How will you do it if you have to scale it up? What is the basis of scale up in this case?
Sir,
We
if
are
it,
not looking at the tip speed anymore. We are not looking at the aeration, or we are looking at the aeration.
sir, if we talk about specifically fermenter for solid state, we can introduce to multiple tray or in a multiple sheets in between to increase the surface area.
Okay.
So this can be introduced in specifically fermenter.
You have seen like that. You have read about it.
Yes, I have read about that we can give the extra sheets or extra trays for to increase the specific surface area for the cell growth.
Okay. You have anything else to add?
Just this stacking we can do like one above the other, and maintaining a 10 to 15 centimeter of space would be enough.
And each and every tray is still porous in nature, so that the media can drip in.
So what?
How do we add the media?
So we can provide a media by dripping itself, or we can supply it through the specific tray.
Okay.
Yes.
Scale up would be interesting thing for me.
Scale
I
up
do like the idea of having multiple trays. You are basically using the same facility. You are increasing the overall, but the volume does not change, right? So you're not changing the volume. You can just changing the spacing between the microbes. I don't know how productive or how efficient that process would be. That's the only concern that I might have. But I like the idea of using solid state fermentation for this. I have read too some papers where they have actually used solid state fermentation for alpha amylase production. So I'm happy that you were able to find some one of those papers to go ahead with. Anybody has any questions for the group? No? So it's like 1,000 to 5,000 per kilogram, 1,000 grams. So that means $1 a gram. Not bad. It's cheap. It's actually very cheap. Good. Thank you. Bluff. Your turn. Bluff is.
Good afternoon, everyone. This is your team Bluff from the morning session. And for the second session, we'll be explaining how we'll be basically the entire bioprocess design for the production of alpha amylase. So I'll be handing over.
I like the idea. You initiate it and you just transfer it.
So, going to the alpha amylase, as we know, alpha amylase is used in various industries, especially food industry, like the nutraceutical industry for controlled delivery of nutrient, like different bioactive compounds, and in food industry in such a way that in breweries, like bakery industries, and other fruit and vegetable processing industry, we are using alpha amylase. And in case of alpha amylase, as you know, like for example, if we are talking about the bakery industries, we have, or in brewery industry, it should have the ability to withstand the temperature. But the bakery industry, the concern is that at a particular temperature, it should be inactivated. That's a major constraint. So accordingly, we have to modify the enzyme, and even naturally, we can see that there are various types of alpha amylase as extracted without genetic engineering from various sources like fungal amylase, and for barley malt they are extracting alpha amylase, and like Bacillus subtilis and Bacillus cereus species, which also produces naturally alpha amylase without even genetic modification, and some other organism also producing. So why we are going for this DNA technology specifically for this extraction and for improving the yield in such a way that we are using Bacillus acidocola. So this is organism which produces heat stable alpha amylase, and our aim is to focus on brewery industries because we are giving a higher temperature, and for the like as we know, alpha amylase is also known as liquefying enzyme, beta amylase is saccharifying enzyme. So the liquefaction is required in breweries to produce like initially we have to produce the specific amount of glucose for the utilization of yeast. So first of all, we have to introduce this enzyme instead of other method, instead of introducing the glucose source as well. So from malt we have to convert it into glucose. So for that purpose we are like our idea is to produce alpha amylase for the same. So Bacillus acidicola, and we are usually it is present in soil. So we are isolating different cultures and different strains will be there. So from that we will be isolating the strains which produces higher amount of alpha amylase and which can grow in like a different variable conditions, and which can produce a definite proportion of alpha amylase. And after that extraction of the gene, we will be identifying the gene and design the primer and like primer for the same, and next followed by the PCR amplification, then cloning into vectors, and we will be specifically selecting the vector for E. coli, that is PET, that 28A plus, the specific vector we will be selecting, and after that it is introduced into E. coli. As we specifically selected E. coli in such a way that even in previous slides you might have seen that E. coli is intracellular production of alpha amylase, even though the production is quite high and this generation time of E. coli is quite lower, so it can produce in a shorter period of time. And so after that, the selecting in the vector and the vector preparation, so digest the plasmid with a restriction enzyme and phosphorylate the vector and to prevent the self ligation. It's followed by the ligation process, ligase the PCR amplification gene into the linearize the plasmid using DNA ligases enzyme, then prepare chemically competent E. coli, then add the ligation product into the component cells and heat shock or electroporate. Like that's the method we are used for transfer the genes, this particular gene. There are different types of like gene transfer mechanism are there. So heat shock is the best method, as we know, like even different types of methods we are using for heat shock. So impulsive electric field can be used for the heat shock in order to create the pores and like reversible pore formation and transfer the gene. So we will be limiting the electricity for the same. So after that culturing, after that the gene expression we have to see like whether it's producing the particular, it's producing the alpha amylase or not. After that test the activity of the purified enzyme to confirm its functionality, whether it's shown the heat stability. Even sometimes it may chance of even already we are using the electric shock, sometimes mutation may happen, we can't say, but it may create some changes in the protein production. So even though we have to check whether it's showing the specific properties or not. So after that the gene expression. Yes. So we will be checking for the same. So after confirmation we will be selecting the particular strain for the so there's a mistake like by editing is the alpha amylase production, not monolith. Okay, title. So title. So alpha amylase production. So so low cost, we'll be using a low cost carbon sources like molasses can be used, but even though like even purification is another matter, so we'll be checking for the same. So the nitrogen source which consists of peptones and ammonium salt, and even though even mostly alpha amylase for food grade it does not require that much purity, but even though like purity is required, but for example brewing and all anyway it's going for distillation, so purity does not create much issue. It's not a big deal in case of brewery industries, but for pharmaceutical grade it is important. And next one is a salt which is commonly supplied sodium chloride and magnesium sulfate, and substrate for the injections specifically we will be using lactose based system because it's instead of IPTG. And fermentation conditions specifically, there are in initial case we'll be following the growth temperature. For the temperature is almost 37 degrees Celsius, and for in case of during induction period it is 30 degrees Celsius. And this IPTG is nothing but isopropyl beta D thiol acetopyranose, and dissolved oxygen is also required. That is almost the 300 to 500 sorry 20%, keep above 20% using, like even dissolved oxygen we have to keep above 20% using air sparging, and agitation is almost 300 to 500 rpm we have to provide, and during induction stage it is 200 to 500 rpm based on the oxygen demand. And the type of fermentation we are following, like either for small scale production batch fermentation, and for large scale production fed batch fermentation. And during harvesting induced OD, like we will be checking for the OD value that should be at 600 nanometer, so it should be in the range of 0.6 to 0.8. In that condition we will be harvesting it. So in case of downstream processing, so as I already mentioned, if it is for, depends upon, once we are focusing on this brewerage purpose or brewery grade, for in beverage industries there is no need of higher purity. So but in generally we have given the procedure for centrifugations at 8,000 to 1,000 rpm, and for almost 15 to 20 minutes. So we'll be separating the pellets of E. coli. Then cell lysis has to be done because it's an intracellular enzyme, so we have to break down the cell. So that is HCl buffer, which is commonly used, and lysozyme, and the sonication, ultrasonication can be also done for the cell lysis for the easier separations of the in cellular components, and we can isolate this protein. We have to degrade the proteins. So meanwhile we have to assure that the alpha amylase should be retained, and after that followed by centrifugation, then purification, and we are following affinity chromatography, that is nickel NTA, that is nickel nitrotriacetic acid, which is used as a resin, and for histamine tagged alpha amylase, and wash the column with low imidazole buffer and elute with high imidazole concentration. If the enzyme lack tags, employ ammonium sulfate precipitation and ion exchange chromatography, and followed by the purification stage. And for that, size exclusion chromatography is commonly used in order to remove the aggregates or other impurities. And for stabilization, so for example, if you want to keep the enzyme for a long time, we have to bind it with calcium plus ion to stabilize it, and followed by the lyophilization and formulation into the buffer. For example, we have to stabilize, the pH should be maintained nearly 6 to 7. And one more thing I forgotten to add in that, E. coli, which produces some protease enzyme, so we have to knock out the gene corresponding to the same. That's it. Thank you. So techno-economic analysis. So we haven't even gone through in details about the techno-economic analysis, but approximate cost for almost we calculated $145 per kg of enzyme, because according to the beverage grade we don't need to purify in details, but if you are going for chromatography and all, then definitely we have to this cost should be increased up to a higher level.
Okay.
But if you are eliminating that stage, we can reduce the cost.
Okay.
Thank you.
Thank you. Now, if I just want to go back, corn steep, liquor, molasses, or whey as a primary substance. Okay. Do you know anybody who can sell me whey for $5 a kg? If you know, please give me the address.
In India, there is.
This is Indian, or you're estimating in dollars?
Generally, we are saying whey is not commonly used in most of the industries. As per dairy industries, even most of the industries, they are just what I understood is, even whoever working in dairy industry, whey is not at all utilized. Only Amul is only producing whey proteins from whey, and even it's too costly. That's why the whey proteins cost too much. That's why most of the industries,
So
they
where
are not
are you
using.
getting those whey protein isolates?
Even in India, only Amul is producing, as per my understanding, because most of the industry, they are just recycling it. Most of the dairy industry in
Okay.
India, they are not utilizing.
Okay.
So definitely, instead of just going for their like
You know that it's not like that they have a, oh, here is the milk, here is the whey, and they can give you the whey in hand. It's like it's drained out
they are like
in the
even
drain.
draining one.
So you will be collecting it from the drain.
No, sir. We can directly collect from them. If, for example, otherwise they have to go for their secondary treatment. They have to
Pre-treatment
up.
of your product, sorry, pre-treatment of your feed stock is always important before you go into your
Because
production.
in dairy industry anyway, after whey will be collected from dairy industry. Even after pasteurizing, they will be going for this like coagulation. So definitely it's pasteurized whey. So no worries, you can take it.
I like the confidence, but I would really like you to pass the FSSAI board then. With that comment, yeah, you just mentioned that it's already pasteurized. Why the hell I read this? No. You don't need to. You have to. Any question for the group? Oh, you have multiple questions. Good. Which part? The recombinant DNA part.
Actually, I want to know how did you incorporated the his tag to alpha amylase?
Wow.
You have not mentioned here any gene sequence or anything.
Oh, you're talking about this one.
Next slide, sir, I guess.
The his tagged alpha amylase. That's not in the recombinant DNA part. That was basically for the affinity chromatography.
Yeah, but sir, they have to incorporate previous variants so that they can use in the affinity chromatography.
How did you do that?
How did you do that?
You have to have the mic to answer that question. It's okay. It's okay. If you don't know the answer, just say you don't know the answer. You can look into it and you can get back
I
to
don't
it.
know exactly the gene editing, how
Okay.
it's done, but even like six histine,
Okay.
corresponding gene corresponding to 6 histine can be introduced along with the primer.
Yeah.
So we can tag it.
Okay. Good.
So I didn't add it exactly in this slide because it's already we discussed regarding this in the previous
Okay.
thing.
Okay.
So
No issues. Any other question? Yep. Procatalysts have a lot of questions because they're a competitor actually using the same technology. Good. I like the competition.
Sir, can you please go to the costing side slide?
The cost $5, $10, $20 for?
Sir, for purification, they use a two chromatography system. One is affinity chromatography, and another is size exclusive chromatography. And using both system in large scale, using multiple solvent, multiple things, how can you manage in $60 only?
Using the NIT facility. That's the cheapest way you can do, right? Because if you have a technology business incubator here, you get free access.
But still, sir, the price of solvent, electricity, it's
Yeah, you convert 60 multiplied by 80.
5,000 rupees.
Right, because you are using US dollars, right? So you're
Only 5,000 rupees.
oh yeah. Then you hire one of the master students to do the project, then it becomes free. It's a very good question. To be honest, this is wrong. I think you just did like, okay, you know what? We have to do this. 5, 25, 25, 30, 60. It's just random values. Like the numbers that you have chosen 5, 25, 10, 15, 60, 20, 10. Those are random values. Like, it will come to your mind. If I was in your point, I would say, okay, 500, maybe 1,200. Cell lysis, maybe 800. You can do that way.
They are bluffing, sir.
But I know, I know. That's the bluff, right?
Yes, sir,
Because
that's
the team
the bluff.
Team bluff is trying to bluff us out. I'm not happy. I'm not happy. With your techno-economic analysis, you get a 0 star. Just for this. Okay, I do have a question. I know this is the last session, so we are having a nice time, but I would definitely love to ask you a question. When you say 200 to 500 RPM based oxygen demand, like RPM for the agitation purposes, what is the size of the bioreactor you're talking about? You need the mic to answer that. Like for the data that you have here is for 100 liters? It's a value that you are estimating, or the source that you used actually had these values?
Sir, for 100 to 120 liters only, we have referred the papers. In that they have mentioned like for E. coli. It's not specifically for the production of alpha amylase.
Okay, just for the cell growth.
Cell growth.
So you're taking the gene from the bacillus, introducing it to E. coli. So E. coli becomes your host. Okay, and then you use the DBTL, which is good. I liked that idea. And so this is for 100 to 110 liters.
Yes, sir.
I still feel 200 to 500 RPM is pretty high. I'm trying to imagine. Is there a fan here? No, we don't have a fan here. What would be the RPM for a fan? Somewhere 100 to 200 RPMs?
150.
So if I take that and put it inside a 100 liter, I don't think my E. coli will last any. They will be like flying everywhere. No, I'm just curious. This 100 to 200 RPM seems a bit higher. I might be totally wrong. You are using the sources. I don't know. So, because you have to mention the resources.
Actually, we don't have practical experience while doing this, so we have only the papers which is available.
No, I agree to that, but it would be nice to have, whenever you're making this type of presentation, it's always nice to have the sources so that we can go back to them and just see if it makes any sense. But I like what you did. Thank you.
Thank you, sir.
Okay, the last group. No name, or you're the group of Mr. Aswath Vishnu? What's the name?
Sir, our group name is Prolase.
So you're still Prolase, right?
Yes, sir. Pro means
PROL.
LAYSE. Combination of protein and alpha amylase.
I like the name, but I'm just saying, like, it appeared like this on my screen.
Okay, sir. Yes, sir. We can start.
Okay, proprietor. We can mention this.
HS, sir.
Okay, good. So production of alpha amylase from Bacillus licheniformis using directed evolution. Nice, good.
So here I have used Bacillus licheniformis. Why means it is a commonly used microorganism for enzyme production, and like it is a natural producer for alpha amylase, highly suitable for hosts, and then it exhibits rapid growth under optimal conditions like 37 degrees Celsius and under a pH of 7.3, and then it can reach high cell density level, density level at QSL. It is for cost effectiveness, and then one key advantage is there. It is like Bacillus licheniformis is there. It is the ability to secrete alpha amylase in the culture media. So it reduces the downstream process cost.
So it's extracellular.
Extracellular. It is extracellular. So it reduces the downstream cost. And then coming back to the recombinant bacteria, like we have taken the strain like CBB0302. It is a strain, and then we have taken the vector PHY AML, and then we just integrated with recombinant technology, and then some preparation processes that like carbon source like sugar, and then nitrogen source like peptone, and then yeast extraction, and then phosphate phosphate group is there, and then trace element is also there, zinc, iron, magnesium, it is there. And then now comes under to the, oh, there is no direct evolution. Morning we have updated the presentation, sir.
This is the only one that we received.
Morning we just updated. We just forgot to add that direct evolution process.
It's okay if you can explain me.
Okay, I will explain it, sir. Like we have to extract the plasmid. I mean, that plasmid is there, no, PHY AMYL, by using what is that? By using alkaline lysis method, and then introduce genetic variation via mutagenesis, using of error-prone PCR to introduce random mutations, and then after that random mutation, we just comes under clone modified.
So you said error-prone
error-prone
PCR.
PCR.
Okay.
Random
For random
introduce
mutation.
random mutation in alpha amylase.
Okay.
By using Taq polymerase which are altered magnesium or manganese concentrations, and then the run around PCR run around 25 to 30 cycles for a mutated variant.
Okay.
And then clone modified genes back to PHY, that plasmid strain, I mean plasmid, by using of some enzymes, restriction enzymes to linearize the plasmid, and then coming back, I mean random mutated gene is there, no, just by using the T4 DNA ligase, we will ligate to plasmid, and then transform the updated plasmid into the E. coli for amplification, initial amplification, and then by using what is that plate transformines, by using LB agar, it is like a lysogen agar broth, by using with antibiotics such as kanamycin, and then extract the amplified plasmid to the plasmid, and then transform that to the Bacillus licheniforms, and then prepare. I mean, again the same process comes, like plating the transformants, and then with selective agars like LB agar with antibiotics, and after that screening method come. Screening is like with starch iodine assay. By using of that starch iodine assay, we will identify the significant starch hydrolysis, like color will change, disappearing of blue, black color. So after that, secondary screening process happen, like testing of enzyme activity, thermostability, and then pH tolerance and catalytic efficiency. First, test thermostability, like incubate enzymes at 70 degrees Celsius for 30 minutes, because it is I am using that Bacillus licheniforms because it produces the alpha amylase in the culture media, so that I am using incubating enzyme at 70 degrees Celsius for 30 minutes. And then by using, measures the residual activity using DNA assay method, and then comes under pH tolerance. pH tolerance, it is test activity in buffer ranging from 4 to 10 by using citrate, phosphate, or tris HCl. And coming back to catalytic efficiency by using performance of Mitchell's-Menton growth kinetics, by using that. Yesterday, I think you informed that VM or KM? VM, Vmax to that one. And then coming back to the fermentation process, we have used first we have used the initial batch, and then fed batch. This is a continuous stir tank reactor, followed by Nikhil.
So the fermentation process we used was a fed batch fermentation process, which was a stir tank fermenter operated in fed batch mode. So we took initial batch phase and then fed batch phase. The initial batch phase was about 12 to 24 hours, which was operated at 7 pH and 37 degrees Celsius temperature. And the next was a fed batch phase, which was operated for 24 to 72 hours. Next, the upstream processing and the fermentation part is done. Then we went to the downstream processing part. The downstream processing, first of all, includes filtration. As he told earlier, that our product is extracellular, that we don't want the cell, our product is in the broth itself. So first of all, we filter the cell and biomass that was all collected in the broth. So filtration, with the help of filters, we filtrated all the cells that are there. And then we went for like filtration and then centrifugation. We want to, like, all the heavier particles, we want to remove them, remove with the help of centrifugation. And then we went for purity ion exchange chromatography, Now we would have gone for affinity chromatography, but that's pricey. So we went for ion exchange chromatography, and our product, which is alpha amylase, is at pH 7 is negatively charged. So we went for a stationary phase, which is positively charged resin. So through ion exchange chromatography, we went for purification. And for concentration, we went for concentration, ultrafiltration, and stabilization. Concentration, again, with the help of like, and concentration, then ultrafiltration to the help of ultrafiltration membranes, and stabilization with the help of compounds which are inexpensive like sucrose. Okay. And then we want the product to be in powder form, okay, which is more lasting. So we want to increase its shelf life. Okay, so we went for conversion to powder form through spray drying, like lyophilization. Okay, and with the help of quality check, and then we went for packaging. Next slide, please, sir. The next slide, the last slide, like explains the costing all of this. So first of all, it includes the capital cost. The total capital cost is 2.1 CR. Now it might look very big. It is because it includes everything. Like in the first session, you told about no one mentioned out the land and etc. So 2.1 CR includes the everything price. Now let's say we are supposed to pay it in 10 years, amortized. Okay. So our cost becomes 21 lakh per annum. Okay. Then the capital cost per kg becomes, we are like operating 50 kilos, 10 kilos over 500 batches over the year. Okay. So the cost becomes 420 rupees per kg. Okay. So operating cost. Then we go for operating cost. Our raw material cost is 14,25,000 for the entire year. Utilities 15 lakh, labor 37 lakh 50,000, maintenance 7 lakh 50,000. Then the total operating cost comes out to be 74 lakh 25,000 rupees. And the operating cost per kg comes out to be 1,485 rupees per kg. Okay. So total cost per kilogram comes out to be 420 capital cost and the operating cost 1485. So our total cost comes out to be 1905 per kg. And also compared with the one available in the market, that was 1990 per kilogram. And that was also for starch processing grade. It was also not good grade like pharma grade. And that was we have prepared. Okay. So if we see the revenue production, so like do the analysis, then in 2.2 years only, we can pay back all the things that 2.1 CR we have assumed. That's it, sir.
Okay, and this 2.1 CR included the R&D
everything.
part also.
Everything, sir.
If I give you that much money, you will work on this?
Sir, obviously it's a theoretical model. If
let
you
me,
give,
let me go
definitely I
back,
will
look
work
into
for
my
it.
bank accounts. If I have enough money, I will come back. We'll invest in
Sure,
this, okay?
Sure, sir.
I like the idea. Now, see, basically, my intent with these two case studies for you was not only to work in a team, you get to work with the multiple people with different backgrounds. But the point is that it's easier to know or learn about a new technology. So I can teach you, let's say I taught you synthetic biology. If I can start teaching you microbiology, I have to prepare a little bit, but I can do that. I'll teach you all the techniques and everything. We have never done that in the lab. You have no hands-on experience. When somebody comes, they give you a speech, they will say like, okay, these are the things that we did, this is how we can approach establishing an industry or developing a product. Product development requires a lot of understanding, not just about the product, but about the process that you have to go through. But it also has to have who's going to be your end user. That's the key. When, let's say, if my industry is developing the straps for the watch, who would be my end user? Is it the public or is Apple? If I am developing the strap of the quality that Apple requires, my end user is Apple. Once Apple buys it from me, what they do is up to them. Their end user is you, the customer, right? So depending upon whom you are targeting, your process development changes. The amount of capital that you are going to invest will change. So having that entrepreneurship is good, but having an understanding of how to approach it is very, very important. Understand the value of your product, understand the market size of your product, only then try to implement it. Location, we call, right, for when you are trying to establish a particular industry. If Rourkela steel plant was not established here, established somewhere else, let's say another 1,000 kilometers away, where they don't even have access to railway or road or the iron ore, they will not be a profitable company, right? It has to be that way. When you are establishing with respect to your feedstock, you have to make sure you have access to your feedstock. Theoretically, in an experimental world, as part of my research work, I can say, hey, I can use agricultural feedstock for production of this, but I have to have the capability of utilizing it on a large scale. If I don't have that capability, the technology is not worth for me. Sima? I hope you learned something from me. This is the last day I'm here. I leave pretty soon, but thank you so much. This will be it.